Thank you and good morning. I would like to remind everyone that comments made today will include forward-looking statements, which may include statements regarding the progress, success, and timing of our ongoing and planned clinical trials, data presentations, collaborations, regulatory filings, and future research and development efforts, among other things. These forward-looking statements are based on current information, assumptions, and expectations that are subject to change. They are subject to significant risks and uncertainties that could cause the company's actual results to differ materially from our current expectations. Accordingly, you are cautioned not to place undue reliance on forward-looking statements. Please review the more in-depth information about forward-looking statements in the slide deck, which is available on our website in the events section, as well as the full description of risk factors that can be found in the documents we file with the AMF in France and the SEC in the United States. Forward-looking statements speak as of today, and Nanobiotix undertakes no obligation to update them to reflect subsequent events or future circumstances. With that said, I'd like to open the call and introduce Dr. Jeffrey Bachman, Oncology Practice Head at Cello Health. Jeff, please go ahead.
Thank you, Kate. It's my honor and pleasure today to be moderating the Study 1100 virtual KOL event. Hopefully the next one will not be virtual. I will be giving a few introductory remarks, followed by an introduction by the CEO and founder of Nanobiotix, Laurent Levy, and then we'll get to hear some of the details around the study, as well as preclinical data coming from Drs. Weiss, Shen, Seward, and Welsh. So without further ado, let me give a brief introduction here. So I think we're all quite well aware that the approval and the rise of the checkpoint inhibitors has dramatically changed the treatment paradigm for many different cancers. I think we're now at something like 18 or so approvals, different indications, lines, combinations, et cetera. And, you know, they are projected to be some of the leading drugs, and Keytruda in particular projected to be the leading biopharmaceutical drug in 2026. So that's a major sea change, and it is around the use of checkpoint inhibitors that much of the ongoing development within oncology has begun to happen. So next slide, please. So although the majority of clinical development is still focused on what we'll call somewhat artificially non-IO agents, IO agents are starting to make up a significant proportion, about a third of the pipeline. And what's important is really the diversity of what those agents are, a variety of agents that are working on innate adaptive agents at the interface of innate and adaptive, targeting different cell types and pathways, all ultimately designed to try and improve the responsiveness of patients to the available checkpoint inhibitors. Next, thank you. But one thing that has been clear over, say, the past decade since the approvals of the first checkpoints has been a struggle to create a novel IO combination. Lots of novel targets have been brought forward. There have been a number of stumbles, some outright failures. Notwithstanding, it's a lot of the recent excitement that's been going on, particularly at ASCO LAG-3, news that's been coming out on TIGIT, some data also on things, co-stims like CD40. By and large, most of the combinations that we see in practice across different therapeutic categories are really combinations of checkpoints with radiation, checkpoints with chemotherapy, or checkpoints with targeted, tyrosine kinase inhibitors. Next slide, please. And so the diversity of those pipeline agents and novel targets that are trying to improve on the responsiveness, both the percentage of patients responded and the durability, the deepness of the responses, is really reflected here in the latest CRI data that just shows the sheer amount of combination studies that are ongoing across the range of approved checkpoint inhibitors. And as you see kind of in the middle of the pack there is the color code. Radiotherapy represents one of the modalities that is being combined with the CPIs in order, again, to improve the responsiveness. And, that represents a very interesting approach and very germane to today's discussion. And one of the important things is that many of these novel agents, including what we'll be hearing about today on R3, involve efforts to modify the tumor microenvironment, the TME, and to try to address some of the remaining cell populations that are enabling the immunosuppressive environment that the tumor is basically when it co-opts the immune system to protect itself. So we'll be hearing about this powerful approach that Nanobiotix has developed that really links together the idea of radiotherapy and the intersection of the direct control that it has, as well as the potential for systemic control through abscopal effects in combination with checkpoint inhibitors. So without further ado, I'm going to pass the baton to the CEO and founder of Nanobiotix, Dr. Laurent Levy.
Thank you. And good morning, good afternoon, everyone. As you have seen in the previous presentation, there are plenty of ways that people are trying to get into the IO world to solve the problem and to try to bring new solutions for patients. They have, most of them, something in common, which is they are targeting a biological pathway or a biochemical pathway. At Nanobiotix, we think that if we want to impact the life of millions of patients, we need to use different approaches, different angles. And that's why we decided to use physics. And we developed this technology looking at what is the most widely used tool in oncology and finding radiation therapy. As you may know, radiation therapy is used in the vast majority of patients getting cancer, around 60%. And you can see here on this slide that most of the big cancer patients get radiation therapy during the course of their treatment. Thing is, you can't cure all those patients just with radiation. You need other things. And some of the issue with radiation could be easy to understand. First, that's a very powerful technique. You can technically kill any type of cell with radiation. The issue is, in many cases, when you have your tumor and you try to irradiate the tumor, it is surrounded by healthy tissue. Therefore, you're limited by the dose you can deliver in the tumor without causing too much harm to the patient. And that's why in a number of cases, you would have an inadequate local control with radiation that will ultimately provide a local invasion of the tumor or a systemic expansion of the disease. You could also have an inadequate systemic control. When you have a primary tumor and some metastasis around the body, then it's hard to irradiate everything. So there is a lot we could do to improve and to change the game with radiation therapy. And we think because it's a practice that is widely used, bringing something in this field could make a big difference for millions of patients. So at Nanobiotix, we have developed a product which is called NBTXR3, which is a potential first-in-class that has a tumor-agnostic component and a combination-agnostic aspect. It is a suspension of crystalline inorganic nanoparticle that you will inject directly into the tumor. And you make a one-time injection before the first session of radiation. So why this has to be nano? Because we need something small enough to go into the cell. And on the top of being nano, we have chosen a very specific material, hafnium oxide, which is a super X-ray absorber and also a very inert material. So with this, we have a product that is injected once that will absorb the energy when inside the tumor and deliver more damage linked to the radiation activation. So how does this work? Here, you see a cancer cell receiving radiation therapy and random damage appearing. If you increase the number of the session or if you increase the dose of radiation, you will increase the number of random damage into this cell. On the right, you see a cell that have received nanoparticles and absorbed them. Every dark spot here, there are hundreds of nanoparticles that have penetrated the cell. And when you send the usual dose of X-ray, those particles will absorb the energy and will deliver around them a high spot of damage. So it's not anymore random. It allows a high spot of damage in very particular places of the cell, therefore creating a lot of damages, and potentially damages that are way different from the radiation therapy itself. So how do we use this universal physical mode of action? First of all, you can use that for the primary destruction of the tumor. Following the irradiation of the primary tumor that have received the nanoparticle, then you will trigger more efficacy and potentially decrease the size of the tumor. In parallel, there is a subsequent biological consequences, which are linked to the activation of the immune system based on the physical destruction of the cancer cell. So you have a local effect plus a systemic effect due to the immune response. This is not anymore a dream. We've been working years to try to develop this product and reach a very important milestone for the company and the product and the patient, which is the first positive phase 3 in soft tissue sarcoma. You can find those results in the Lancet Oncology that have been published in 2019. And basically what we have shown here is that the trial has met both primary and secondary endpoint, and showing that when you had nano enhancer on the top of radiation therapy, then you improve the outcome in terms of efficacy. So that was the initiation of the development of this product. Now we have a very focused development strategy. We want to bring this product on US and European market and now Asia for head and neck cancer patient. And the phase 3 that it will start soon is in locally advanced cancer patient. And that's the primary focus of the company. 2, and that's why we are here today, we want to establish this product as a pillar of immunotherapy. And we will see one of the first data we have generated in human today. And 3, with the help of our partner, like the MD Anderson Cancer Center or the recently partner we signed partnering with Lian Bio, we want to expand the use of this product across oncology. That's why you see that we have a pipeline that includes a number of clinical trials with a clear focus of the company within the head and neck area, both as a single agent with radiation or in combination with checkpoint inhibitors. And that's what we are going to talk about today. And on that, I will give the mic to Dr. Jared Weiss to talk about the IO world.
Good morning, everyone. So I am a clinician and clinical trialist, and I'd like to talk to you about how I view the landscape, the unmet need, and how R3 is attempting to fill it. Going forward a slide. And going forward once again, this is the inspiration that has occurred from PD-1 checkpoint immunotherapy. You're looking here at data from Keynote 177 in MSI-high colorectal cancer. And what you can see here at the right of the curve in blue is what we call the tail of the curve, what for very appropriate reasons is all anyone can talk about. These are patients who for the first time with metastatic cancer are achieving durable control and perhaps even cure for a proportion of patients. And of course, Pembro in blue handily beating chemotherapy in red. This is not a one-off story for MSI-high colorectal cancer. You see at bottom similar data for second-line PD-L1 positive metastatic lung cancer that regardless of dose in blue or red, pembrolizumab is handily beating chemotherapy, and of course doing so with a significant minority of patients achieving durable control. And next to that, very similar data for melanoma. Going forward a slide. We see that many patients, however, are left out in the cold. When we look at the Keynote 48 data, randomizing unselected patients with metastatic head and neck cancer to pembrolizumab or the standard chemotherapy plus cetuximab, we see that not only is pembrolizumab failing to beat the standard regimen, but that durable control is really being achieved for very, very few patients. This limitation is not a weakness uniquely of head and neck cancer. You see below it similar data for gastric cancer, and you could show similar curves for just about any human cancer. As a consequence, there have been multiple withdrawal of accelerated approvals based on these limitations. Going forward again. So I want to talk about the shape of the curves here. And I want to talk about a theme that we've seen from the early mirroring data with the PD-1 inhibitors through the phase 1s all the way through the phase 3s. The shape of the curves, the exact cut points, vary a little bit from tumor to tumor and context to context, but the theme is identical from pretty much all solid tumors, which is that there are 3 proportions of this curve that describe differential benefit. Going forward one, please. We can see that the majority of patients are what we might call primary resistance. The left side of the curve is a cliff, meaning that either on the first assessment or since all of these trials allowed you to go to the second because they use IR resist, by first or second scan, the majority of patients have progressed. And this is the inconvenient truth that no one wants to talk about with PD-1 immunotherapy and is the unmet need, that the majority of patients do not benefit. The second group is similar in theory to chemotherapy, which is to say patients that achieve benefit but subsequently lose it. And going forward one more, we'll see the final group, the inspiration, those who have durable control. Now, how big of a problem is primary resistance and secondary resistance? Well, let's go forward a slide and look. You're looking here, once the slide comes up, at data for patients with an indication for checkpoint inhibition. So we're not looking at patients who are completely ineligible. And what we see is that of these eligible patients with an indication, only 13% overall actually achieve a response. So this is not a small unmet need. This is not the footnote or caveat. This is the problem. Going forward a slide. One of the dominant ideas out there was to combine radiotherapy and immunotherapy. And you can look at this in two ways. You can add checkpoint inhibition to a standard radiotherapy, or you can add radiation, particularly hypofractionated radiation, to a metastatic PD-1 paradigm, and both have been tried. The far left, you're looking at data from the Pembro-RAD study. What this study did was randomize patients to a standard cetuximab and radiotherapy, or a competitor, pembrolizumab and radiotherapy, for locally advanced curable head and neck cancer. And what you can see at a glance from each of the three curves is that whether you look at survival, progression-free survival, or local regional control, there is no advantage to the substitution of pembrolizumab. A similar effort was conducted with avelumab, where it was added, in this case, to standard of care chemoradiotherapy. And what you can see is that there is no significant improvement in outcome as measured by either survival or PFS. Now, what about flipping that on its head? What about using radiation to synergize, to improve the efficacy of checkpoint inhibition? This is a particularly important graph to look at together, the one at the far right, to address whether the data you're going to see later with R3 is really just related to the radiation or whether you need the R3. So this experiment was done. You're looking at data from metastatic head and neck cancer or incurable head and neck cancer, looking at a randomization between nivolumab alone or nivolumab with SBRT. And what you can see at a glance is absolutely no improvement whatsoever in survival. So that experiment has been tried, and unfortunately, it did not work. Going forward one more. So what are my unmet needs as a clinician? When my patients recur, there are three patterns that I see. I see local regionally advanced disease only without curative modalities. So the patients had all the surgery that they can have or are willing to have, all the radiation that they can have or willing to have. I can draw a line around the problem on a scan or in some cases on physical exam. And yet, despite the disease being local or local regional only, I'm asked to give systemic therapy to control it, to control their pain, their trouble swallowing, whatever the local effect is. That's very frustrating because systemic therapy has real tolerability issues and, of course, doesn't have a significant cure potential. The next third of my patients have both a local regional problem and a distant problem. And the final third have a distant problem only. And so what you could imagine, and I'm sorry I didn't make a figure for this, is overlapping Venn diagrams where 2/3 of my patients have a local regional problem and 2/3 of my patients have a distant problem with overlap. And so as a clinician, I need a new therapy that addresses both. Going forward a slide. And so the more mundane hypothesis behind R3 is the simple destruction of the local problem. This local destruction is tumor agnostic. You're going to have that amplification of radiotherapy to fry local tumor no matter what tumor you have. In addition to that, you subsequently trigger multiple biologic pathways that can prime for an immune response. As you see at left, these mechanisms are pleiotropic and therefore potentially broadly applicable. And so in summary, I have a high unmet need for both local control and distant control, and R3 is a promising approach to attempt to do so. Going forward a slide. With regards to the immune stimulation, I want to share with you an analogy that I sometimes make with my patients. In this analogy, a hot tumor or a bonfire is our analogy for a dramatic immune response. And the unfortunate reality is that most tumors are cold, as scientists call it, which we might make analogy to here as an unlit log. Now, if you pour gasoline on an unlit log, going forward one, all you get is a wet log. You don't get a fire, no matter how much gasoline you pour on it. You need something to start that fire. Going forward one, R3 attempts to start that fire. It attempts to be the match or the spark, if you will. And if you do start a small fire, if you initiate it, then gasoline like PD-1 inhibitors such as Keytruda, going forward one, can create that bonfire that we seek. And so with that, going forward a slide, I hope that I've framed for you the context in head and neck cancer and solid tumors in general. And I'd like to now pass the microphone over to my colleague, Dr. Colette Chen, to walk you through the data that we've produced to argue how far R3 has come in showing that it can do that.
Thank you, Dr. Weiss. It is my pleasure to present for you some of our early data from the study 1100. If you can go forward one slide, please. This study is a phase 1 study of intratumoral NBTXR3 injection in combination with anti-PD-1 in patients with advanced cancers. This study is being conducted across multiple institutions across the U.S. We have been treating multiple patients on the study at UNC, Dr. Weiss and I, as well as several other sites. Dr. Seward as well, one of our speakers, has also been treating patients on this trial. Next slide, please. This study evaluates this NBTXR3 activated by radiotherapy in 3 cohorts of patients. The first cohort is patients with inoperable locoregionally recurrent or recurrent and metastatic head and neck squamous cell cancer, treatment of recurrent disease in a previously irradiated field that's amenable to reirradiation. The other 2 cohorts, cohorts 2 and 3, are patients with lung or liver metastases that are not in a previously irradiated field and are in any primary cancer type that's indicated to receive anti-PD-1 therapy. In all 3 cohorts, patients may be anti-PD-1 naive or be anti-PD-1 non-responders. Next slide, please. So just to review these inclusion criteria, for PD-1 resistant patients, they must meet criteria consistent with either anti-PD-1 primary resistance or secondary resistance. Patients who have had prior anti-PD-1 therapy undergo washout. These patients undergo SBRT following NBTXR3 injection. The dosing is site-specific, so patients with head and neck recurrent cancers receive SBRT to 35 gray in 5 fractions, lung metastases 45 gray in 5 fractions, and liver metastases 45 gray in 3 fractions. The primary endpoint of this phase 1 study is the recommended phase 2 dose. The secondary endpoints, objective response rate, safety and feasibility, and body kinetics, and exploratory endpoints of survival, duration of response, and biomarkers of response. Next slide, please. So to go over the timeline of this study, patients are injected intratumorally with NBTXR3 on day 1. On day 2, NBTXR3 is confirmed, visualized in the tumor, typically as part of the radiotherapy planning scans. Radiotherapy begins usually 1 to 2 weeks following NBTXR3 injection. This lasts over 3 to 5 fractions, as discussed previously. And then anti-PD-1 administration begins the day after radiation finishes. Importantly, this timing is consistent with data from the Pacific study that suggests that beginning anti-PD-1 therapy within 14 days of radiation derives that increased benefit. And the other thing that I'll point out is that there is a dose of steroid that's given with the NBTXR3 administration on day 1. And subsequently, that this means that there's at least typically 2 weeks between that steroid administration and anti-PD-1 initiation to decrease the impact that steroid may have on the efficacy of anti-PD-1 therapy. Just to review a little more depth, the patients that have been treated across these cohorts, there have been 16 patients treated thus far, 7 in the head and neck cohort 1, 6 in the lung metastasis cohort, and 3 in the liver metastasis cohort. You'll note that most patients have head and neck cancer as their primary tumor type. You'll also see that prior anti-PD-1 therapy varies across these patients. There are multiple anti-PD-1 naive patients, the majority of those in cohort 1. And then there are also several patients, in particular those in the lung and liver metastases cohorts that have primary or secondary resistance to prior anti-PD-1 therapy. The PD-L1 expression level also varies. I will just point out that there are several patients who are PD-L1 negative, and those patients, in addition to those who are PD-L1 positive, have shown response. As you can see in the best observed response in pink, the majority of patients have had at least stable disease as their best observed response, regardless of prior anti-PD-1 exposure or PD-L1 expression level. And then you'll also see on the bottom row that the majority of patients have had no serious adverse events from this treatment. Next slide, please. And then to review the adverse events in a little bit more detail, the adverse event profile has not differed in type or grade from what is typically expected with radiotherapy or anti-PD-1 agents. You'll see that there have been very few grade 1 or 2 or 3 plus adverse events across either of the dose levels that have been studied so far. I will note that there was one patient in the head and neck cohort 1 that was anti-PD-1 naive who did die from pneumonitis about 2 months after NBTXR3 injection. He was anti-PD-1 naive, and this was thought to be related to anti-PD-1 therapy, possibly related to NBTXR3. Next slide, please. Looking at these graphs of best observed response by RECIST, on the left, distinguished by primary tumor type, and on the right by previous exposure to anti-PD-1 therapy, you'll see that of 13 evaluable patients, 10 of these 13 have had clinically meaningful tumor regression regardless of prior anti-PD-1 exposure. These will include patients who have had stable disease or partial response or complete response, and you'll see that we actually have had 2 patients with complete response. Next slide, please. Another way to look at this tumor regression data, you can think about clinically meaningful tumor regression, which is these yellow bars. Clinically meaningful defined as at least 10% regression in all target lesions from baseline. Or we can look at investigator-assessed objective response per RECIST. We'll see that across all 13 evaluable patients, there's been over 75% clinically meaningful regression, and this is maintained whether patients are anti-PD-1 naive or anti-PD-1 non-responders. The investigator-assessed objective response per RECIST is also in the 50 to 60% range. All of these response rates are much higher than what's typically seen with standard of care with delivery of immune checkpoint inhibitor therapy alone. I will also note that, as I think you'll see in a little bit later in this presentation too, that there have interestingly been at least two patients who have had a response distant to the injected irradiated lesions. And so we are seeing some very interesting systemic responses to this therapy combination of radiotherapy, NBTXR3 injection, and anti-PD-1. I think if we move on to the next slide, I'll introduce our next speaker, Dr. Seward. Dr. Seward's the director of the Head and Neck Cancer Oncology Disease Group at Johns Hopkins, and he'll be discussing a little bit more of this data in depth.
Good morning, everybody. I hope you can hear me. It's my great pleasure to be part of this and to present some of the exciting early data from the 1100 study. I was involved in designing the study, and it's really rewarding to see now some of the early data coming through. And Dr. Chen already provided you a high-level overview of some of the early results. However, I wanted to specifically highlight some aspects that I think are really remarkable and unique about NBTXR3. Just for background, when I first heard about R3, as I call it, which is the short version, I thought this is really interesting. This is really strange. This is a hafnium oxide nanoparticle. I've never heard of such a thing. I knew that this was used in lasers, etc. But why inject it in tumors? And then people started telling me that sarcomas that are, completely immune cold, as Jared pointed out, become inflamed. And I was very intrigued by that. And so that was actually the starting point where I realized, well, this has really, it's not just a radio enhancer, but it is actually potentially an immunotherapy. And that was the basis for the study, 1100, to combine it with a checkpoint inhibitor. Next slide, please. Can we move on to the next slide, please? Wonderful. This is essentially the same data that Colette already showed you, and I just want to summarize it. Here you see an overview of anti-PD-1 naive patients. So you can argue, well, PD-1 may have activity anyway, but it is interesting to see that out of 5 patients, 4 patients had tumor regression. Arguably everybody had tumor regression. And we saw 2 very deep responses and 3 patients who had a resist response. So by any standard, if you think about lung cancer, head and neck cancer, the response rate for PD-1 is maybe around 20%. So here you have 3 out of 5 patients, 60% achieving a response, which is good. But again, small numbers, you can say interesting, let's wait for more data. But it is interesting, especially deep responses, as you know, are associated with long-term survival. And so it's nice to see these deep responses. However, what I think is particularly remarkable is looking at anti-PD-1 non-responders. And this is what is shown on this slide here. And that's where I think most people start saying, this is interesting. And this really, I think, should catch our attention. So here we have 8 patients who are PD-1 refractory. And this was well defined in the protocol. And you can see here, and these patients should not respond to anti-PD-1 because they're refractory. And here you see that 6 out of the 8 patients who are PD-1 refractory had tumor regression. And importantly, 50% had actually a resist PR or a resist response, including 1 CR. So, the green one is a deep response. I would say 2 or 3 patients had deep response. Again, those are responses that have a good quality that oftentimes are durable. You also see the blue patient here who had kind of this up and down pattern, which is an interesting feature of immunotherapies, which we see. We don't see this with chemotherapy or radiation as commonly, but it is a classic feature of immunotherapy. So overcoming anti-PD-1 resistance, in my mind, is a large unmet need. I call it the holy grail. What comes after PD-1 failure? And at least, again, this is a small number of patients, 8 patients, but it seems to be rather robust. Next slide, please. The other piece that I think is interesting, when we first introduced PD-1 inhibitors, people started talking about the so-called abscopal effect, was a distant effect, and I think we'll hear more about this mechanistically from Dr. Welch afterwards. But, obviously, if you irradiate one lesion, that lesion will shrink. But what about the lesions that are distant? Patients most of the time will die from distant metastasis. And so there was an early on description that abscopal effects are systemic responses. So tumors respond outside of the lesion that is treated. Abscopal effects, unfortunately, with radiation and anti-PD-1 are exceedingly rare. I always joke that, in the typical practice of PD-1 with radiation, there's more people studying the abscopal effect than there are actually patients with abscopal effect. So in fact, I recently wrote an editorial in JCO, saying that abscopal effects are really not that common and really are more anecdotes. However, what really, again, caught my attention in the 1100 study, here we observed out of a small number of patients, multiple abscopal effects. Now, I want to walk you through this. So on the left side, you see a patient with lung cancer, stage 4 lung cancer, and you can see here in red, the red curve is the NBTXR3 injected lesion, and blue and green are lesions that are non-injected. Obviously, well, we're not surprising that the red lesion shrinks, but then blue and green also shrink, which is a classic abscopal effect. In fact, I have never seen an abscopal effect in patients. I've treated hundreds of patients before joining the 1100 study. And then you can say, well, once in a while you see this. But then look at the second patient, so the second one from the left. The red lesion actually gets a little bit bigger, and this may be a flare, or maybe this is tumor progression, but what is really intriguing is the blue lesion and the green lesion either stay stable or shrink, again suggesting a systemic effect. The same is true for the third patient where the red lesion is injected, and then you see a little bit of an increase and then a decrease in the green lesion. Again, it's early data, let's wait and see, but it's very intriguing that you see these systemic effects and kind of this undulating pattern, which is classic for immunotherapy. And then finally, the patient on the right, which is a head and neck patient, this is a patient who's PD-L1 negative, and in those patients, PD-1 only has a response rate of 5 or less %, and we don't give pembrolizumab single agent in those patients, at least not in the first line. So the red lesion is injected and stays stable, but look at the blue lesion, which is non-injected, non-radiated. That's a classic abscopal effect. So just to summarize, we have multiple abscopal effects in a small study, while previously abscopal effects, at least I have never seen one using just PD-1. So there's a signal. Again, I think we need to collect more data. But if this is true, I think this is, in my mind, the first study that reliably induces abscopal effects in patients. The other part, and I think what I just told you, I think is highlighted here. I don't think we need to spend much time here. The left patient is a clear abscopal effect. And then you see this kind of flare with initially an increase on the right side and then a response. Again, very typical for immunotherapy. Next slide, please. So the other part I wanted to show you is this delayed response and this undulating pattern. Oftentimes, with really good immunotherapy responses, the responses take time to evolve. We've seen this with ipilimumab, where a lot of the durability comes in late. We've seen this with some of the other agents. Deep responses take time to emerge. And we see the exact same thing with NBTXR3. You can see this on the right. It takes several months to get, 200 days to get to a complete response in this patient with head and neck cancer. And you can also see this kind of up and down pattern on the left side. And then it took a biopsy to get to a path CR. So sometimes radiology does not reflect fully what you see, but pathology confirmed that this was a path CR, which is something very special. Next slide, please. So on this slide, you see a so-called Swimmer's plot. Here you see a total of 7 patients who are PD-1 naive. You see a complete response at the bottom in green, really durable, almost 2 years. And then you can also see that 4 out of these 7 patients continue on study, which is encouraging. In blue, you see partial responses, the triangle. So, I think encouraging in PD-1 naive patients. Next slide, please. Here you see the PD-1 refractory patients. Again, those are the ones that I really, really feel compelled by. This, I think, is 9 patients. And here again, you see complete response in green. This is G, cohort 3, patient G. And you also see 3 patients with partial responses in blue. Again, for patients who have a poor prognosis, who are anti-PD-1 refractory, seeing such results, I think, is very encouraging. Next slide, please. I would like to summarize. When you look at, this is data from the head and neck KEYNOTE-48 study. And this kind of repeats the slide that Jared showed previously. When you look at categories of patients, obviously there are some patients that respond to anti-PD-1, usually about 16 to 20% in head and neck cancer. Here we have 3 out of 5 patients responding, which is good. Again, small numbers, we have to be careful. We'd like to see more patients, but it's encouraging. But in my mind, what is really intriguing is the second group, patients who have reduction in tumors in non-injected lesion, and that is unique. And really, I think the upper category, that's where I think we're seeing incremental benefit. Again, a small study, but certainly encouraging. Next slide, please. So let me summarize. NBTXR3 administration is intratumoral, and it was feasible, and it's actually very well tolerated with patients. It's more of a logistical challenge. But we can work this out. This is done frequently. We're good at this. And side effects are minimal locally from R3. The side effect profile, hence, is very tolerable. And side effects are driven really by the standard of care modalities such as radiation, in this case, SBRT, and the PD-1 agent. R3 does not seem to add meaningful side effects. 10 out of 13 patients seem to have benefit and response with tumor regression. In my mind, there's a strong rationale to combine with immunotherapy. And what I think is very compelling is that even prior IO exposure, being PD-1 refractory, does not limit benefit from this particular agent. So in my mind, it is encouraging both in PD-1 naive patients as well as in patients who have progressed on anti-PD-1. So I hope to have shown you some unique aspects of these data. And really, in my mind, while R3 was developed as a radio enhancer, I believe that really major potential is in combination with immunotherapy. This is really an immunotherapy, and in particular, the abscopal effects are unique. And with that, I would like to pass on the baton to my colleague, Jim Welsh from MD Anderson, who has done some beautiful data in preclinical model that actually underline some of the clinical observations. Jim, handing it over to you.
Great. Thank you for that background and introduction, and excited to be here and to talk to you about some of the mouse data that we're getting of how nanoparticles can be combined with immunotherapy and work systemically. Next slide, please. So really, a lot of the work, you've seen the rationale of how nanoparticles can be put into a tumor and enhance radiation and local control. But what can we do beyond that? Most of our patients die from systemic disease that's not controlled. And so can a nanoparticle control that? Can we do disease and treat areas that are beyond that? Next slide, please. So here, what you see, this is a mouse model, and it's just nanoparticles with radiation. And we have two tumors, one on one side that's treated with radiation. But what's really interesting is the untreated tumor that you see here. And so I want you to focus on that. And so when we irradiate one tumor with the nanoparticle, we get better local control, not surprising. But what's really interesting is the tumor on the other side that's non-treated has a response, which is quite an interesting phenomenon. And you see that translating over here in terms of survival. Next slide, please. So the middle panel here now shows treated tumors, and then on the right side, untreated tumors. And in the middle are the CD8 immune cells, which we know are important for the immune response. And so again, you have all the controls here of radiation, nano by itself, and then the nano with radiation. And it's great to see that the CD8s are increasing, the tumor gets irradiated, which is what's leading to better local control. So nano is really working as an immunotherapy here, which is wonderful. But what's interesting is look in the unirradiated tumor. This tumor doesn't have nanoparticles, it's not irradiated, and the CD8 cells go up. So you're getting immune cells from the first tumor that's treated that go systemically into the other tumor. So really, we're all familiar with COVID and COVID vaccines now. And so what's happening here is this tumor is becoming a vaccine and providing T cells that go into other tumors to provide control, which is really a neat benefit to show that nano is really kind of like an immunotherapy, as the medical oncologists have pembrolizumab, the radiation oncologists now have their first kind of immunotherapy. So it makes sense now to see if we can combine this with the classic immunotherapies like PD-1. So tumor cells can escape the immune system by expressing surface receptors that act as stop switches for blunting the immune response. And drugs like anti-PD-1 and PD-L1 can block this and allow the T cells to respond again. So we wanted to test checkpoints with nanoparticles and radiation. Next slide, please. And what we decided to do is to test this in a very challenging model. And next slide, please. And so this, we could, of course, test it where PD-1 works, but my lab has a model of PD-1 resistance. So it's a pretty nasty tumor, and it's now resistant to PD-1. And we decided to test nano on that. And again, we have the primary tumor and the untreated tumor. I'll call your attention to the untreated tumor. And the green line here is now the separation, showing when one tumor has nano and is treated with radiation in a PD-1 resistant model, the PD-1 drug now works better systemically. So we're able to address checkpoint resistance in this model. You see a benefit in survival and lung metastases as well. Next slide, please. Now, this is really nice data because, again, we're starting to learn that this nanoparticle really works as an immunotherapy in multiple levels. So we did nanostring analysis that looks at various immunologic pathways. And these are triplicates. So you've got the green, which is control on the left. In the middle is XRT and PD-1, a treatment we know dramatically can improve outcomes in patients. And then on the right is when you add the nanoparticles. And it's very clear to see that the red is the increase in these immunopathways. So we have green, and then a dramatic increase, but it's in almost every immunologic pathway nanostring looks at, which is really quite surprising. So it's antigen processing, dendritic cell function, macrophage function, NK cell function, T cell function. All these important aspects in the immunoprocess are getting improved when just one tumor has the nanoparticle and gets irradiated. Next slide, please. So this slide will now break down the difference of like the B cell improvement function. So in the black area here is just kind of control. The blue is PD-1 and radiation, which is our standard in the clinic. And then the green is when you add the nano. And the top middle one is a T cell function, CD8 T cell function. And then the bottom left is dendritic cell function. So improving two of the most important components for immune processing and function. Next slide, please. So what we wanted to do next is to see if we could go beyond PD-1. So this is a PD-1 resistant model. It's great that nano makes PD-1 start working in a PD-1 resistant model, but obviously in the clinic, we're not going to give someone a PD-1 drug if they're already resistant. And there's many new exciting second generation immunotherapies like LAG-3 and TIGIT. So what we wanted to see if we could add these second generation immunotherapies on top of nano to get a further response compared to what we were getting right here. Next slide, please. And again, I'll draw your attention to the curve on the right of the secondary tumor. The one that gets irradiated gets great control with nano. The checkpoints help get better control. But the tumor without nano is now responding much better when we add these second generation treatments. And you can see that in the orange curve versus the black curve separating. And then now for the first time, we're seeing long-term survival. So at the bottom curve, we have mice that are totally cured and they have no more, cured and their tumors are not growing and they're able to survive an extended period of time, which we're seeing for the first time. Next slide, please. So now that we had mice that were surviving a long period of time, we wanted to do the next work on immunology to see if there's memory produced. So just because it worked short term, will it keep working? Did these CD8s get activated and keep working and keep surveilling to reduce new cancer cells that come up? And so here we took the mice from this experiment that were treated, we re-injected them with a large amount of tumor cells directly into the mice, and no tumor was growing. So it's kind of interesting to see the curve because you see the 4 animals that got tumor and what it looks like. And the very bottom line with those black dots, that's the curve. There is no growth. So these mice truly have memory now. And you see that in the lung mets as well. They do not produce any lung mets. So what this shows you is first we showed that nano in a tumor helps control the tumor. Wonderful. Next we showed that nano in a tumor when irradiated controls other tumors. So it's treating other tumors that are not injected with the nano. Very interesting. Now what we're showing is nano radiated in tumor is now controlling tumors long-term, producing memory. So even though the nano's done its job, the radiation's done its job, when new cancer cells come up, they're addressed by the T cells and cleared, and so they're not going to produce metastases and recurrence. So it's very interesting how nano is working immunologically. The last slide here on the bottom left, you can see this is looking at central memory for the CD8s in the blood, and then it's control, and then with nano. So proving again that we're increasing central memory. Last slide, please. So in conclusion, I'll go through that Nanobiotix has an anti-tumor immune response that triggers an immune response and an abscopal effect independent of checkpoint inhibitors. So it works even without checkpoints on board. Certainly adding it makes it better. Adding NBTXR3 to radiation and anti-PD-1 significantly improves the abscopal response. The combination therapy of NBTXR3, XRT, PD-1, LAG-3, and TIGIT significantly promotes proliferation of the CD8 cells, improves tumor control, produces long-term anti-immune response and memory, and increases survival. The anti-tumor effect of the same combination is heavily dependent on CD4s and CD8 cells. If we knock those cells out, it does not work. So it proves it's an immunologic mechanism. The surviving mice from the group treated with the combo are then re-injected, and they don't produce any tumors and able to control those tumors. And lastly, the combination therapy maintains a significantly higher percentage of memory CD4 and CD8 cells, as well as a strong anti-tumor immune activity that then control the tumor, resulting in long-term anti-tumor immune response. So with that, it kind of gives a perspective of where we're going in the future, how this can work for future trials in combination with immunotherapy. And with that, I'd like to hand it back over to Jeff to take us on. Thank you.
Great. Thank you, Dr. Welsh. All right. So I get a visual where I can see everyone and we can have our virtual roundtable. All right. So we've got some questions coming in. A couple of them actually are relevant to the very first question that I want to put to everyone, and that is, in terms of the 1100 study, how are you really seeing the data in the context of previously reported relevant comparable studies? And then we can dive into some specific questions that people have about the 1100.
So Jeff, it's okay. I'll start out.
Yes, please, Dr. Sir.
So I think the first thing to say is that this is a small study. So it's just a few patients. So you always have to be cautious with small studies. However, and the other part that I think is another disclaimer is it's a mix of patients. I think this is, 3 different cohorts. We have lung cancer, we've had neck cancer. So you also have to keep in mind that this is a study that has a mix of different histologies. But with those 2 disclaimers, there is something very unique about this study, which is activity in PD-1 refractory patient. And that by itself, is something that is always very attention-grabbing at any meeting. And I think that's really unique. And that really, if you have a 50% response rate, again, be careful, mix of patients, small numbers, but that's unique, and very special. And I think that really stands out. So I'm not aware of another agent that has had a 50% response rate as an immunotherapy in prior PD-1 refractory. So that's the first comparison point. The second comparison point is with prior studies in lung cancer, in head and neck cancer, generally speaking about a 20% response rate. And we do know that deep responses are meaningful. So I think it will be very important to see as we gain more patients on the 1100 study, do we continue to see these deep responses? Because ultimately, we would like to see a survival benefit. And I think deep responses, depth of response is very meaningful as a correlate of ultimately survival. So that's something I would watch for. Again, with a lot of caveats, this looks quite promising. The other thing you can compare this to is there are other agents, in development. There's monalizumab in a phase 3 study in head and neck cancer, has about a 30% response rate in PD-1 refractory, but that's with cetuximab, so it's a bit different. And they're not as durable. It's not maybe an immunotherapy. There's now LAG-3 that's being developed, and there's a number of other agents. So in my mind, this looks favorable in comparison to some of these other agents. And there is something that I think is also important. So if you think about, TIGIT, LAG-3, it seems like these work in PD-L1 high tumors. So they already work in inflamed tumors. R3 is such a different agent because in my mind, nobody has done anything like this. It changes probably the microenvironment by killing the tumor cells in a way that radiation doesn't. So I think it's a very different entity. It's not another checkpoint. It's not chemotherapy. So there's something very unique. I would use as different from some of the other agents. So not only are the data good, but I think there's also something that's at least important to study because of the different characteristics.
Great. Anyone else want to comment on that?
Yeah, this is such an important question. I'd like to highlight and underscore a few points that Tanguy made and add to them, because I agree with every word. But in terms of benchmarking this, it's easy for the naive patients to say this is just what PD-1 does. And so as Tanguy was speaking, I pulled up the numbers from the randomized study of pembrolizumab versus chemotherapy to benchmark this numerically for everyone to say, what do we expect? In the most highly selected patients, those with a high CPS PD-L1 score, which in head and neck is over 20, with 133 patients, the response rate was 23%. That's the most highly selected group. When you get down to the PD-L1 intermediate, it goes down to 15%. And for the PD-L1 negative, who we showed responses in, it gets down to 5%, right? So that's, these are very small numbers. There should always be caution with very small numbers, but it's better for small numbers to look very good than very bad. If you're going to compare with those caveats in mind to the historic expectation, the numbers are extraordinary if shown in larger studies. And then in terms of the pretreated, it's important to look at how pretreated a patient is. So if you take a patient who's had immunotherapy and at their first scan has progression, if you keep them on immunotherapy, there is a response rate to that second scan. Whether you call this pseudo-progression, whether you call it delayed response, I don't want to get into today, but there is a real response rate, right? If you look at the OAK data in late-line lung cancer, patients heavily selected by their practitioners to continue had a response rate in PD-L1 despite prior progression. And that's why we showed you the individual patient level data, including duration of prior therapy on PD-1 here. Some of these patients with responses at non-radiated sites and prior progression in PD-1 had actually gotten it for quite a fair amount of time, meaning that any response subsequently observed was unlikely related to simply treatment past progression. And so when placed in that context, I think, again, with the caveat of the small numbers, this looks quite extraordinary. The other thing I wanted to say about competing agents is that I'm not sure they're actually competing at all. I think Dr. Wells has shown you some nice data on why this can combine with some others. Looking at monalizumab, I think its strength is potentially actioning patients with loss of HLA. And that actually could synergize nicely with R3. If you bring in NK activity while you're potentially in parallel increasing antigen expression, as Dr. Welsh has shown, that could potentially work nicely together. So I actually don't see competition so much as additive, or one can fantasize perhaps even synergistic benefit. And of course, extensive preclinical and correlative work will be needed to show what the optimal combinations with R3 will be.
Great. Thank you. Thank you, Terry. Colette, any comments? Or?
Yeah, when Dr. Weiss was talking about these heavily pretreated patients, I just thought I would highlight with one of the patients we've treated on this study. He had had either chemotherapy with immunotherapy or just immunotherapy alone for at least 6 months prior to treatment on this study with progression, and he had disease at multiple sites. He had an enlarging lung mass that was starting to cause him symptoms. He had distant nodal disease at a AP window node as well as an inguinal node, very distant from his lung site. And when we started him for treatment on the study, his initial scan, before we even did the radiotherapy, actually showed progression of the lung mass. So we were very concerned. This is one of these patients where you're worried that he's already kind of blown through the standard immunotherapy, chemotherapy combinations. His primary tumor is growing. He's symptomatic. Typically, you would think that we just really don't have much in the way of options for him. So once he got treated on study, NBTXR3 in the lung mass, radiotherapy, and then checkpoint inhibitor, we were just really, really pleased to see that the lung mass shrunk, which you could say could be from radiation, but both the AP window and distant inguinal nodes also responded. So this was just something that I don't think any of us would have expected normally.
Great, yeah.
And the nice thing about the nice thing about this treatment is that it helps both the local disease. He was having symptoms from that, but it also works distantly. So very,
Great.
very, very promising.
Which patient number was that?
That was patient number 5. I think it was one of the graphs that Dr. Stewart showed.
Okay. Yeah, because there are some questions about that before I get to specifically. So I think we've addressed one of the questions as well within that general response, which was kind of what would you like to see as a more mature data set in terms of overall response rate or duration of response. I think the discussion of kind of that Jared mentioned of kind of the benchmarking there versus what you're even seeing in the small data set, gives a large window for improvement. But maybe we can rephrase it slightly differently and say, what is the minimum you would hope to see that would be meaningful to you with this agent on the supposition that, as data matures, that might start to decay in terms of kind of what you've seen to date in terms of the high response rates?
Yeah, I think maybe this is a little bit callous, but I think any activity that's real in PD-1 pre-treated patient that has a flavor of an immune response, I think is clinically meaningful if it's durable. If you put a number on that, I would always say at least 10%. I'd like to see 20%. We're far above that in the small numbers here, but I think activity, immunotherapy-like activity in PD-1 refractory patients is very, very meaningful usually. So upfront, ideally, that's where you have, Jared, quoted the numbers ranging between 23 and 5%. On an average, it's about 20%. So as long as you're above 30 or 35% upfront in PD-1 naive, that's also meaningful. The final point, which I think Colette made it actually very nicely, but I really, really appreciate that point. In head and neck cancer patient, the big challenge is if you have a big bulky tumor and a couple lung mets, the big bulky tumor in the neck is where patients have symptoms and pain and bleeding, and PD-1s are not ideal for that. So it's not just the activity, it's also that this is a treatment that has a particular effect on a large, bulky local tumor where patients really need some sort of treatment. So that's oftentimes where clinicians feel like PD-1s are not ideal anyway, because they might take a long time and they really don't do anything for the local disease. But here you have a treatment that has this additional local effect. So not only is it activity, but it's also that local effect that, at least for a subgroup of patients, is for a clinician very attractive.
Yeah, Jim?
Yeah, great comments. Agree with all that. As you guys were talking, there's one point that I kind of wanted to bring up as well. Is clearly the future is combination of multiple immunotherapies. PD-1's exciting, but it's not enough when we have to put multiple agents together. And the challenge as we do that is really toxicity. You're hitting multiple mechanisms all at the same time, inducing inflammation throughout the whole body, and you don't want that. You want it just in the tumor. And I think that's where nano has a very unique advantage, because this is an inert piece of metal. It's an inert nanoparticle, and then gets activated once you hit with the radiation. And so, when you're thinking about a future where you're going to be combining multiple checkpoints and multiple drugs with a nanoparticle, I have found it's a really nice combination because it's safe, it's non-toxic, right? It only gets activated when the radiation turns it on, and you're not having overlapping toxicity where we saw things like when we did Ipi-Nivo, we saw more depletion of Tregs and colitis. So a lot of companies are reticent to combine their drug with an immunotherapy because they don't want your drug to cause a toxicity, that's going to get blamed for. But with a nanoparticle, it's quite different. So I think you get this nice immune benefit without really a high risk of toxicity. So
yeah, I think that's exactly why we kind of refer to things like this as orthogonal for exactly the reasons you've mentioned. So, Jared?
Dr. Welsh read my mind. I was going to talk about toxicity too, right? We had very little toxicity outside of what you expect with PD-1 alone. In terms of high grade, a single hyperglycemia event, a single pneumonitis event, a single soft tissue necrosis event, which of course is paying taxes on your lotto earnings. But these are exactly what we see with PD-1 alone. And of course, you can never benchmark efficacy without benchmarking toxicity. I also wanted to simplify this a little bit because I think we're talking from the perspective of clinician scientists. And I am cognizant that this is mostly an investor audience. And so I wanted to make the more simple statement, which is that if you look at our minimum bar for adoption, and where the data is at right now, you're probably at a multiple. There's a lot of room for regression to the mean before you start to fall below the threshold. This is really rather extraordinary relative to our minimum benchmark.
Great, thank you. And maybe I'll use that because one of the interesting questions, and this has been touched on both in terms of the clinical data and Jim, your preclinical data, and I think this exists, throughout the community, is confusion around abscopal effect and what exactly is meant. So the specific question is actually, given the 1,100 patients are all on a systemic checkpoint, is it appropriate to classify non-irradiated lesion shrinkage as an abscopal effect?
So I think there's a lot of controversy around abscopal effects. And, the original report in the New England Journal of Medicine, or not the original, but one of the high impact reports from Sloan Kettering, there was question, is this really an abscopal effect? Because it may have just been a delayed response from ipilimumab. However, in this study, these are patients who are PD-1 refractory. So the systemic effect from the PD-1 arguably is not there because the patients are PD-1 refractory. So any systemic effect from the non-radiated lesion, I think it is appropriate to call an abscopal effect, or as Jim likes to call, just a systemic effect. And maybe that's more appropriate. I always would caution, if you see one, maybe it's just dumb luck, But if you start seeing multiple, it becomes much more convincing. So in my mind, seeing two or maybe three, depending on how you interpret it, you see these smaller, tumor regressions in some patients, then you had two very clear cases of an abscopal effect. Again, I've treated hundreds of patients. I've not seen a single abscopal effect, and I've tried. And, I always say radiation doesn't do a thing for abscopal effect, and maybe Jim disagrees with me on that. But the way I've done it, it hasn't worked. But now on 1100, there's two patients where it worked. And I think the difference is not the radiation. I think the difference here is really the R3 enhancing the radiation. And it's just different. This is not radiation anymore. This is a very high dose local. It's very unique. This is not just radiation, but it really is a local effect. So that's my personal perspective. Yeah.
As you were talking, I was thinking about, people kind of know the immune cycle, right? This cycle that goes 1 through 6, where it starts with antigen release, antigen presentation, priming T cells. So when you give just a PD-1, you're assuming that cycle is already done and you're helping the exhausted cells to work again. And then that's not enough. But when you give R3 and radiation, it really starts the cycle. It starts that whole immune cycle. You cause this massive antigen release, you prime a new crop of cells, and then the PD-1 has a chance to work again. So I think mechanistically it makes sense why it can address PD-1 resistance, because it's causing a new cycle of T cells to get stimulated that then attack the cancer. So.
Yeah, and certainly I see lots of early stage companies who, in their experimental models, actually do show, abscopal effect, but again, it's always in the background of the necessary systemic derepression on a checkpoint, right? So,
yeah, and Jeff, can I just make a point? We can cure
please.
tumors in mice all the time. We can make abscopal effects in mice all the time, but this is actually abscopal effect in patients, which again.
Absolutely, yeah.
Very, very unique in that regard.
Absolutely, yeah. My point was simply that to think that somehow, you wouldn't see an abscopal effect, or that the definition is somehow dependent on not having a checkpoint doesn't make sense, right? So, all right. So now there's some very specific questions around the 1100 study. One of this is, I think, the first question that actually came in, and first it makes reference to, and I think you did address most of this in the presentation, but just to make sure that this is clarified for the questioner, clarifying the 3 new patients, they're talking about comparison from the last data published in November 2020, what cancer types and who are they, clarifying the nature of the patient who died from pneumonitis, as well as clarifying the status of patients J, M, and N, in particular M and N, in which it's said in the question there is no real response or small tumor regression, and what might be the explanation for that.
So let me try to start addressing some of these. So for the patient who had the grade 5 pneumonitis, that patient was a head and neck cohort patient, prior anti-PD-1 naive. And the timing of when he developed pneumonitis was pretty consistent with what we might see with checkpoint inhibitor therapy. I think it's hard to know, I think in any of these studies with checkpoint inhibitor, we're going to see some high grade, potentially fatal pneumonitis or other similar. It's hard to know if NBTXR3 kind of compounds this, but I would say, so far in 16 patients, the adverse events otherwise have been very minimal and very consistent with what we would see with checkpoint inhibitor. So to me, because this patient was anti-PD-1 naive, the timing was very consistent with what we would see from a toxicity from checkpoint inhibitor. I think that was very, very likely the cause of his grade 5 toxicity. I think that regarding the question on, I think, patient J, I believe that was the patient that had a response greater at a non-injected lesion than the injected lesion. And I think probably one of the questions is, why did the non-injected lesion have more of a response? I think this is maybe a result of a lot of times in the injected irradiated lesions, we can see either kind of a pseudoprogression or even just some scarring and inflammation following treatment. And it's hard to, when we do these measurements, we kind of have to use our best judgment. And a lot of times we'll still see whether it's, a kind of a T2 enhancing signal in an MRI that may not actually be active tumor anymore, but just kind of an inflammatory or scarring response. I think that's probably what the case is in the injected lesion. And I do think that the uninjected lesion is demonstrating a distant response, for lack of a better word, an abscopal response. So I do think that's what's happening with that patient.
One other quick comment on Jay before you move on.
Go ahead.
And please correct me if I'm wrong, but if I'm not mistaken, that was also a PD-L1 negative patient, which is important to consider in benchmarking this. When you treat PD-L1 negative patient with PD-1 alone, your response rate is about 5%. So having a response in a non-irradiated lesion in a PD-L1 negative patient is a surprising and favorable event.
Yes, that patient is PD-L1 negative. Mm-hmm.
Yeah,
that's right.
Which is interesting. I mean, I wholeheartedly agree with Colette with your comment about radiology being just not reliable. Especially with immunotherapy. People have resected residual lesions, and then there's no tumor, so it's a pass CR, but by radiology, it's barely a response. The other point about patient A, it looks like a really good response. Like a deep response in the distant lesion, which again is not what you expect in a PD-L1 negative patient.
So, and then any comments on M&N in terms of, explanation for why I know this is the hard question, but, why maybe there's no real response or minimal response?
I don't know that we actually can explain that, except to say that, until one day when we cure cancer, which is the ultimate goal here, right? There are some patients who are cured with PD-1. We can look back 8+ years to the phase 1 and phase 1 expansions of drugs like Nivo and Pembro and see that there are patients with Christmas tree PETs, which is to say everything lighting up, who approaching a decade later are still no evidence of disease. They're probably cured of their disease. And if they're not cured from a human perspective, we've certainly achieved something quite a bit like it. The current benchmark is that we don't do that all that often. And so I don't know that it's really necessary or that we can explain the failures here, except to say that this drug doesn't cure everyone, and we never thought that it did.
Yeah, and I'm reading between the lines. I'm just wondering if it's a reference to, has there been any correlative work that might suggest, nanostring work, et cetera, might say, oh, there's something going back to Jim's comment about the Chen and Millman, cancer immunity cycle, or something about that patient's makeup that says, oh, it's X.
Yeah, I don't think we're going to be able to analyze every patient with tissue. That's a common theme in trials, actually, but I was just looking at N and M particular. So if I look at this correctly, one of the patients, they're only 2 months in. So it's oftentimes, too early to say what the true response, how deep is the response, because these responses evolve late and you just need more time. And at least patient N continues on study. So I think we should give it more time also to see if the response evolves. And again, even in the most favorable patient, what Jared kind of alluded to very strongly, and I want to emphasize, somebody who is PD-L1 100 and has a very high mutational burden, like everything is perfect, only 35% will respond. And so even though the biomarkers are great, the majority still don't respond. So we can't expect everybody to respond, and the biomarkers can only take us so far.
The question is insightful and important for us as a research group to investigate, because we've spoken about the potential to combine. And I do think that given the toxicity profile we've seen, as well as the pleiotropic mechanisms that we've looked at, this is a very combinable drug, if you want to call it a drug. But I think the question is actually rather insightful, because more sophisticated understanding of the answer will allow us to know what to combine with, right? What are we not covering that something else could?
Yeah, and I think that
Jared.
So go ahead, Dr.
Yeah, Jeff, sorry if I'm taking the discussion away from what you were trying to get to. But Jared's comment about, is this really a drug? It's actually a very good comment. Obviously, it's a drug, but it is very different. This is not your average checkpoint inhibitor. And I think also, Jim's comment, this is not something that generates inflammation. This is a physical mode of modality that just kills tumor cells in a very different way, and probably has an effect on the microenvironment. So all the comments about this being complementary, I think, are very important, because this is just different. This is not a me to another checkpoint. This is not another LAG-3 TIGIT. In fact, Jim's data suggests it's perfect to combine with multiple checkpoints. So I think the potential as we develop this further is really unique, because this seems to be very different. This is not something where another company will just say, oh, we'll develop a drug like this. This is not so simple.
Yeah, and I think the question also is germane to the data, the preclinical data on the LAG-3 and TIGIT combinations, because that often, and what Jared said, hoping to have a rational approach to deciding why you use a particular combination in which patients and which, particular cells within the microenvironment you're needing to affect in a given patient in order to improve their responsiveness. There are two related questions I want to put out, slightly different, but still ultimately related. One is about kind of what would you see as the kind of the ideal positioning for R3? In other words, I think they're getting at, would this be seen to be moved forward as checkpoints are trying to do and are doing in terms of going into adjuvant settings, earlier lines? And then another question about kind of usage is, what about the ability to use, IR-guided delivery to go into less accessible tumor types with direct injection?
Yeah. I'll take that first one a little bit about the setting and where you go. And, I lead the immunoradiation initiative at Anderson. So we think about how to get immunotherapy in everything we treat, every tumor type. And, the way I think about it is, we have stage 1, 2, 3, and 4 for cancers, right? And every tumor we have could benefit. So stage 1, you need increased local control and make sure it doesn't metastasize. I'm kind of thinking about lung cancer, but, nano can help there. Stage 2, you've got a little bit more advanced disease. Sometimes we do take them to surgery, but that's a tumor that also can benefit from local control and reducing systemic. And then as you go through the spectrum, stage 4, this is where it's being tested in some of the trial data you see here, where ironically you're using it to provide a systemic boost. So, I think every flavor, stage 1 through 4, needs either a lot more improvement on local control or systemic. And this is an agent that gives you both. So you can kind of tailor it based on those applications. So I think it goes into a lot of different tumor types in different stages as well. So it's pretty universal. And the one last thing I'll say is I'm a radiation oncologist, and it's kind of like the med-oncs have had all the cool toys for a long time, right? You've got the Pembros, the CTLA-4s, you got the TIGITs. It's like, we need something. We need a cool toy.
You're just jealous.
And finally, we have something that, we can control a little bit. You can kind of control where it gets injected, and it makes our radiation take it to the next level. And I'll just quickly, this wasn't the question, but it does something very interesting too. Like if you have tumors that have previously been treated and you can't retreat them because there's too much dose to the spinal cord, we have a real challenge. And we do fancy things like protons, but this is a particle that can now help with retreatments. And that's kind of a whole area, a class with not a lot of competition as well. Different from immuno, but this particle does a lot with radiation and can help the rad-oncs in many different settings and tumor types. So I'll stop there. Thank you.
Yeah, I agree. I think those thoughtful comments. I like the comparison with protons. There's so much stuff about protons, but Jim, sorry, protons are just radiation, and they are like a small incremental change, a little bit better. And sorry if I'm dishing it out on protons, but this is very different. This is like a completely different entity. And protons are, I think it's a very nice comparison. I really like that. Protons have small incremental benefit, but this is potentially, if it really pans out the way it looks, may really change things. Jeff, towards your question, investors love this question. How do you move this forward? I agree with Jim that you can develop this, obviously, but I would say, first, really show it to overcome resistance in the last line. And once it's very solid there, then all the other things will follow. So I'd be a bit cautious, to speculate too much. But in principle, this is an immunotherapy in my mind as a radio enhancer in addition. it would apply to all tumor types where immunotherapy and maybe even non-immunotherapies right now have a role. We saw this data in the Lancet Oncology of sarcomas that in the translational data become inflamed. So maybe there's a potential beyond just the typical, lung cancers, head, neck cancers, bladder cancers.
Yeah, and maybe we'll use that to get to one of the last questions, maybe the last question, which is, other types of combinations, which you were just going to, and Jim, showed the LAG-3 TIGIT data. The question proposes FLT3 ligand in terms of effect on, DC, dendritic cells, but, there's certainly a large range of potential, development stage combinations that one could envision. But kind of doing the thought experiment, aside from the TIGIT and the LAG-3, are there other combinations, whether they're IO or otherwise, that each of you think might make interesting combinations with R3? Jared.
Yeah, so I'm happy to take that. I find it interesting that Mona was referenced earlier, because I think that's actually one of my answers. When you look at the TRACER-X data, and if anyone on hasn't, they're the best thing to come out of Great Britain since the Beatles. What they've done is sorry, that was funnier in my head. Anyway, what they did was they biopsied multiple sites in cancer patients over time to look at the evolution of tumor and immune compartments. And they've shown that one of the dominant mechanisms to resistance to both the modified and unmodified immune system is loss of antigen presentation machinery, particularly loss of heterozygosity for HLA. So, if you think of R3 as lighting a fire, as causing immunogenic cell death, dendritic presentation, things that action that loss of antigen presentation machinery, in my opinion, might be highly exciting to combine with, because then you're really covering all the mechanisms, or most of the mechanisms, of failure of initial response or subsequent resistance. So Mona would be one. Mona is one of several agents that can activate NK cells, which are great at killing cells that have stopped presenting HLA. The other might be CAR T, cellular therapeutics, because you bypass the need for presentation in the context of MHC. So that might be where I might fantasize about going, but I think there's a lot of basic work to be done first, right? The simple stuff of showing that you can add to definitive radiotherapy-based approaches and cure people, further proof of principle that you can add to systemic PD-1 therapy and enhance that activity. And then once you're jogging at a good clip, then you can start to put cure into the rangefinder and try to say what's the last steps to get there. And these are the things that I would personally speculate upon.
Great. I just want to go ahead. Yeah.
Just really quick, some of the things I think about, and I really like the fire analogy that you use, so I'll take that one step further. When we look at all of our trials, the people who don't benefit are the people with low lymphocyte count, right? If you have a low ALC, you don't have the lymphocytes, it just doesn't work. So how do you fix that? And so the fire analogy is, if you don't have the wood, right? There's no wood in the fire, or maybe just a little. And so how do you add more wood? Well, I think the interleukin drugs, the IL-2s, the IL-15s, that can stimulate those immune cells are really nice here. And so we haven't talked about that yet, but you take a patient with low ALC, this is a way to get those CD8s, those NKs, those cells there, and that could combine, and that would make nano better. So I just wanted to throw that in.
Yeah, well, I think some of the chemokines too yep.
Yeah. And those drugs are starting to take off. We're seeing a lot more there, so that's exciting.
Yeah, I'd like to second the comment about the interleukins, interleukin 2, 7, 15. So they're really interesting. And there's a number of drugs coming forward that are interesting. I agree with that. I mean, the other thing that I think we didn't touch on is, there are other intratumoral agents out there. There's STING agonists, there's oncolytic viruses. But all of these have had very modest, data. So, but it's not unique. I mean, I think this is different from some of these other intratumoral agents.
Great. All right, I think we're getting close to the end here. Maybe I give each of you a chance to make any kind of final comments about, R3 and the 1100 study and, what you see kind of going forward. So start with you, Dr. Chen.
Yeah, no, I mean, I think like everyone's been saying, this is early data, but very, very encouraging. I think these are responses that we just would not expect to see. I think, typically this is a group of patients with very advanced disease. They're patients with recurrent head and neck cancer, which typically we have challenge controlling both local and distant disease. These are patients with lung and liver metastases. So, I think one of the things too is that typically we expect these patients to continue, not only to have progression of the disease we can see, but also develop additional metastatic disease. And I think one of the things that this combination is doing is not only addressing disease we can see, but addressing microscopic disease that they likely already have. And so the fact that we're seeing response of 50+%, both in what we can see, but also not seeing any development of new lesions, which is what I would typically expect for these patients, is really encouraging. And the fact that this can be used regardless of the tumor type, as long as it's an injectable area, in combination with IO, I think is very promising. I think one of the other questions that I don't think we addressed earlier was, should we try to access other areas that are maybe a little bit more challenging? I think there is promise there. I had a colleague that was asking me, there was kind of a retroperitoneal sarcoma mass, and I thought, well, that's probably beyond what's typically done right now. But I think we can be thinking a little bit more outside the box, because this is very promising so far with what we've seen. So I'll pass it on to someone else.
Great. I'll go clockwise on my screen. So, Dr. Weiss.
So I do think that you need a logical progression moving forward, right? So as an clinician investigator, I'd love to do everything at once, but in my fantasy world, Nanobiotix has infinite resources. Of course, that's not the real world. So I think a logical development step would be first proof of the clinical principle that we've provided strong evidence of today. So I'd love to see greater patient numbers to prove that R3 can improve the cure rate in a disease that's typically locally advanced, like head and neck cancer. Subsequently, I'd like to see larger patient numbers. Again, it's a little mundane, but to prove more the hypothesis that R3 clinically is improving the response rate and durability of PD-1 inhibition. But then while you're doing this work, you can get biopsy specimens and blood specimens to understand the strengths, better understand at a more granular level mechanism of action, and perhaps actually most importantly, understand the weaknesses to answer the question that we've started speculating about here, which is what can we best combine with, right? Because I think when you look at metastatic disease, it's easy to say, okay, we want a hazard ratio of 0.8. Okay, we want to be greedier. We want a hazard ratio of 0.5 for survival. But I think we have to come back to what patients want. Patients want cure. And philosophically, of course, once that's mentioned, everyone nods their heads. Yes, we want to cure patients. And so I think we need to look at the warts and say, well, what's getting in our way of cure, right? That's the next step. Understand what we need to combine with to get beyond the small but real numbers of patients we're curing with PD-1 now. You could argue what that number is. I would say it's less than 10% in the populations that I'm seeing. So how can we move that cure rate from 10 to 20 to 50, ideally one day in our fantasies to 100% of patients? And I think we need to understand what we're not covering right now.
Great. Dr. Welsh.
Okay, great. So I'm going to give like a historical perspective, right? So radiation's been around for about 100 years. It came from Madame Curie, discovered radium. And in that last 100 years, it's just amazing advances. We're so great at local control, stereotactics, so many advances. It's wonderful to see what we've done since Madame Curie came up with radiation and helped us harness it for helping patients. And I really see this as the next big thing in the fact that, we were all about local control, but we need systemic control to cure patients, right? That's the thing radiation doesn't do, is systemic. And maybe it's no coincidence that Laurent is also a Parisian, right, and came up with this next advent. So now 100 years later, we're changing radiation from local to systemic therapy. And that's only going to happen once in history, right? And I think what you're seeing from this clinical data is that's what's happening here. Radiation is now entering into systemic control. And I'm very excited by that because of the potential of what we can do to help our patients, right, and bring it into a whole new era. So, it's just, now the combination with PD-1s and checkpoints out there, it's really a great time for radiation to start pushing the boundaries, making people think different about it. And I think Nano's a great example of integrating in with a classic installed treatment that's integrated all over the world of radiation. And then this is a treatment that hopefully we can change and improve outcomes for people all over the world with kind of a low barrier to entry, if you will. So thanks.
Great, thanks. And finally, Dr. Seelig.
Yeah, I get the tail end of the stick here, and
we heard the tail end of the curve was good.
This. Jeff, I think that was your decision actually. But anyway, I'll make this short and sweet. The point that I think Colette made is very important. This is small numbers, and while very encouraging, small companies generate small numbers that are exciting. But in my mind, there's something unique about this. And if I were, Nanobiotix, and I think they are thinking about this, how do you get this to a quick approval, given how promising these data look? And I think the answer to that is generate larger numbers similar to what you have right now. And you already have a study going. So in my mind, can you get a data set that is potentially, has potential to go to the FDA to say, we show activity in PD-1 refractory patients. That's a large unmet need. That's where you don't need a randomized study. I also agree with Jared. You should generate the data. There is the head and neck upfront study to show, in combination with radiation. That's a randomized study. But in my mind, this is a hypothesis-forming data set right now that is very promising, but it's small numbers. How do you get to a data set that you could potentially go to the FDA? And in my mind, these early data, if they hold, look very promising for that. And then you can do all the other things, biomarkers, additional combinations, but be laser focused on how to generate a data set for the FDA.
All right, great. Well, I want to thank
I
you and thank the whole Nanobiotix team, and hopefully one day, we'll get to that 100% Jared was referring to. Maybe even before that, Nanobiotix will get a Google Doodle combining R3 and Madame Curie. So, all right, do I turn it back to Laurent or Kate, or?
Thank you, everyone. So I hope you enjoy this event. Thank you for the speakers, and stay tuned. Talking about bigger number, we should see the next update on this trial by the second part of this year. So waiting for you to show you that. Thank you very much, and I wish you a very good day.
Thank you, everyone. Thank you. Have a good day.